Hello, everyone, and welcome to the Praxis webinar, Advancing the PRAX-222 EMBRAVE Study and Other SCN2A Treatments and How Your Participation Matters. I'm Karen Utley, and I am the patient advocacy lead for Praxis Precision Medicines, and I'm so happy to be a part of this team. For those of you who don't know me, I joined Praxis in the end of July, and I am new to Praxis, but I'm not new to genetic epilepsies. I have a daughter, Samantha, who's 15, and she's diagnosed with CDKL5 deficiency disorder. And just because of my own life experiences, I'm truly honored to be working as an advocate with Praxis and to be able to help keep focus on our families and our children. So with that, I am going to start a screen share to get us started. Again, thank you all for joining. We appreciate you taking this time and listening in to this session. So my team that is joining us today to actually give this presentation is our Christian Rubio, the Vice President of Patient Advocacy, Brian Pfister, the Vice President of Global Medical Affairs, and Kelly Dalby, the Associate Director of Natural History and Diagnostics. At Praxis, we are a team of neurologists, geneticists, scientists, and parents of children with developmental epilepsies, working together on one of the largest portfolios of targeted epilepsy programs. And with that, I will get us to today's exciting agenda. We are participating in this program today as a team dedicated to bringing precision medicines to kids with SCN2A, and to share our excitement about the progress being made in our pipeline to serve you. For today's exciting agenda, Christian will be providing a general introduction of Praxis and an overview of our commitment to the epilepsies by guiding you through our study pipeline. Brian will be presenting on the EMBRAVE study for PRAX-222 and the initial dose cohort trial design. And Kelly will be sharing how the SCN2A community participation in the citizen data collection has helped inform the clinical trial efforts for PRAX-222, as well as the opportunity for US families to participate in the new citizen cohort. We recognize that you may have different interests, concerns, and understandings of the terminology that we use today. We will do our best to communicate in simple and clear terms. And if there's anything you don't understand, you can put a question into the Q&A section on your screen. Please keep in mind that you may have some questions today that we can't answer, but because we're still in discussions on some things with the FDA, but we will communicate in the most transparent manner that we can to answer all the questions that we have the answers to right now. And with that, I am going to turn it over to Christian.
Thanks, Karen, so much. And thank you, everybody, for joining us today. Before we get into the full program, I'd like to kick off by sharing some status updates on our 2 clinical stage programs for SCN2A-DEEs. First, a brief update on PRAX-562, our small molecule anti-seizure study candidate, which is a novel sodium channel blocker for rare genetic developmental and epileptic encephalopathies, or DEEs, like SCN2A. We're excited at the prospect of this molecule being designed specifically for DEEs. While the potential for enhanced efficacy and improved tolerability relative to existing anti-seizure medications. To get a clearer understanding of where we are with this study, we've provided this very typical drug development path for a potential new drug. We are now at the end of phase 1, having studied PRAX-562 already in 130 healthy patients, adult patients, and we are now intending to start phase 2 in early onset SCN2A-DEEs and SCN2A-DEE patients outside the United States this year. In parallel, we're working to resolve the current clinical hold with the FDA in the United States. So to be clear, This is not a pivot away from the United States, but rather a parallel path forward to ensure we continue to be able to make progress on this study in the coming months. So I'd also like to provide a status update on today's main topic, which is the EMBRAVE study of PRAX-222, an antisense oligonucleotide, or ASO, which is intended to address some of the most severe symptoms and genetic underpinnings of SCN2A mutation with the goal of truly modifying the disease. So pictured in this slide is not the typical drug development path for a study. Instead, you see what's sometimes called a seamless study, where due to the nature of an ASO, the way it's administered and the highly personalized nature of the medicine, it forgoes the traditional phases of drug development. Instead, this approach, which has been cleared by the FDA for this study to commence, will move from its preclinical state to dosing actual patients in an initial cohort. This seamless path acknowledges both the urgency and leans on a solid history of ASO trials to get the study medication into the kids who need it most faster without sacrificing safety or quality. We'll be starting with an initial pre-qualified small cohort in the United States in the coming months. And simultaneously, we're exploring ways to initiate other cohorts as well. Now, for some of you, you may know Praxis from the events where we've joined you this year and in times past, as well as other community updates. But for many of you, you may be newer to your diagnosis or only know us by some of these updates that you've seen through the community. I want to take a moment to introduce ourselves as a company and to introduce more about our team dedicated to working for and with you. Now, what you see here on our mission statement, I don't really need to read the slide to you. So instead of reading it to you, I want to call out some keywords to explain a little more about what we aim to accomplish faster and more effectively than has ever been done before. This is what drives us each and every day. And in recognition of that, and in recognition of what these communities like yours are going through, and for too many of you in your own homes, we know that we are serving patients with devastating conditions and with urgent unmet needs. A lot goes into developing a drug. It takes passion, it takes focus and trust between scientists and physicians with us, but it also asks the most of you, the parents and the kids, looking for better options. It takes your commitment and trust, and we never take that for granted. Why we do what we do? Well, this is what drives us each day. It's the needs are urgent. We are dedicated to bringing forward new treatments to patients and families to help treat CNS disorders or central nervous system disorders with high unmet needs. And when parents are measuring seizures in minutes, hours, days, we know we need to pursue the science, the regulatory process, the engagement with you with that same sense of urgency, thoughtfulness, quality, and intention. Epilepsy is core to what we do here at Praxis, starting with Steve Petru's work connecting genetic insights from his time at the Florey Institute and translating that science into potentially disease-modifying treatments for kids with genetic epilepsies. And Praxis has parents of children with genetic epilepsies as founders and leaders, not just as advisors, joined by experienced epileptologists with a deep background caring for your kids through their diagnostic and care odysseys, and committed professionals with their own very personal reasons for dedicating their careers to working to alleviate the burdens of central nervous system disorders. Next slide. Now, of course, coming back to you through conversations with you, we've learned so much about your children's conditions and how they're cared for. More than just seizure control, you need solutions to help your children develop as they should and make important gains in self-care and communication. Being able to tell you what's wrong or what they need, or to go to the bathroom by themselves or feed themselves would make life so much better for them and for your family. Right now, there are no medicines out there that are made specifically for your children. Existing anti-epileptic medicines were made for seizure symptoms, but often come with intolerable side effects and don't address or limit progress in other needs you have in neurological development and improved quality of life. Since these medicines were created, the science around epilepsy has evolved, and we know so much more about their genetics. What we're working to harness is precision medicine approaches. We believe that precision medicine approaches will transform the care of rare genetic epilepsies. And we believe that a precision medicine approach offers new potential for treatments that are truly made for your child's epilepsy, with enhanced efficacy and improved tolerability, with true and more lasting disease modification and better seizure control, leading to better lives and developmental outcomes. Next slide, please. At Praxis, we've built this deep expertise in the genetic of epilepsy into the rest of the company's work as well. And our portfolio applies that precision medicine approach, blending small molecule therapies and antisense oligonucleotides that target the underlying genetic cause of disease for epilepsies. And though we have a very strong focus on epilepsy solutions, our expertise in epilepsy is actually inspiring and informing innovation in other central nervous system disorders as well. We have a promising candidate for essential tremor that you'll hear more about in the coming months, as well as one for Parkinson's. There's one clearly of most interest to you, which is PRAX-222 today, and for early onset SCN2A and other DEEs. And Brian Pfister, our medical affairs lead for the epilepsy program, is going to take you through updates and a deeper explanation of it. Following his introduction and walkthrough of PRAX-222, Kelly Dalby, our associate director of our natural history and diagnostics lead for the epilepsy program, will be taking, and an SCN2A mom herself, will be taking you through how your participation in data collection through the Ciitizen platform has helped lead us through so many innovations to get to this point. So without further ado, I'd like to turn it over to Brian and to learn more about the EMBRAVE study.
Excellent. Thank you, Christian. Incredibly excited to be here tonight. I will try to keep my passion and excitement manageable, but it's incredibly exciting in general for us at Praxis to talk about the EMBRAVE study design. As he mentioned, I'm on the medical affairs side. I do have a son with epilepsy, not SCN2A, but I do have a son with epilepsy. So anybody who was here for the first webinar component I had done, it's me again. Thank you for coming back. But I do want to share on the next slide what the first cohort looks like. So Christian was talking about, what the seamless design looks like, and so we'll show the first cohort. So EMBRAVE, how does the EMBRAVE cohort work? Good question. What we have set up is the goal of the first cohort is strictly safety. So it'll be over 20 to 21 weeks. We'll be studying the PRAX-222 in patients with SCN2A. So it'll be open label study. So what that means, everybody will get active drugs. So nobody will get placebo. Everybody will be active. Everyone will know that they have active therapy. And we're getting all these things set up. So as I mentioned before, and as the team had talked about, we're sharing this in real time with you as this is coming out. So we're still finalizing some of the details. So dosing will be in the next few months that we'll get going on that. What we have is in the prior to dosing, the patients have up to 8 weeks of baseline and screening observations. So this allows us to ensure the patients meet inclusion criteria, making sure that they have everything lined up to be able to go smoothly through the dosing and the safety component of the study. What's really important to look at and to focus on here, having worked with other splicing modifiers, having worked in other areas in my past history, we want to do it right. We want to do this right the first time. We want to take our time to really look at this carefully, to understand this, all aspects of safety as we move this product through into the next phase of the clinical trials. So what you'll see is PRAX-222 is going to be given intrathecally. So as before we had talked about, that's going to be similar to a spinal tap injection of PRAX-222. So you'll get one every month. And then in between, there's going to be 4 weeks of safety monitoring. And in between those 4 weeks, there's going to be a 2-week in-home safety monitoring. And so what we're going to do is we're going to capture all of the safety data, all this information, and we'll submit this to the FDA for review and an analysis to get approval to move forward into the open label phase. So as you can see, there's 4 doses, 4 weeks in between each of the doses, with monitoring going on continuously. So we're excited about this. We think that this allows us to create a trial that shouldn't have to stop at the end of the last dose, that we should be able to move into the open label extension. Really, that's the goal that we have behind this design, which I applaud. I think it's a fantastic idea. On the next slide, we'll go through what the inclusion criteria look like. So everybody always wants to know, can I participate? Does my child qualify? So I'll go through some of the main criteria. Basically, patient needs to have, or the child has to have SCN2A variant categorized and characterized, and based upon the genetic testing, confirming that they actually do have the SCN2A variant. they need to have experienced seizures within the first 3 months of age. And the reason behind this is to assess gain of function. So typically what we've seen is gain of function patients with SCN2A variants experience seizures within the first 3 months. The age inclusion is 2 to 18 years. The weight is at least 10 kilograms. And then for the seizure history, they have to have at least 8 countable motor seizures in 4 weeks prior to screening, and then confirmed in 4 weeks prior to baseline observation. So this means that everyone knows how many seizures the child's having at home. We just want to make sure that the patient is meeting that criteria of the 8 countable motor seizures. And then the other important one, I think this is really important for most families to look at and pay attention to, they can be on no more than 2 sodium channel blockers. Because as we noted before, the way that this molecule works, the ASO, is going to be downregulating the mRNA, which is going to encode for the protein for sodium channels. So we want to make sure that they're only on 2 sodium channels at most, and they need to be stable for at least 1 month prior to screening on the sodium channel combinations that they have. And they need to remain stable throughout the course of the trial, because we're not changing, we're going to be changing the sodium channel expression. And so we need to make sure that we're not changing the sodium channel blockers, which are acting on those channels. So on the next slide, everybody wants to know, how do we find out more information? How do we get connected? And if you're interested, please email us at embravestudy@praxismedicines.com. This will allow us to get in contact with you, to be able to give you update information, to be able to set up opportunities for you to participate, should your child qualify. So that's the short and sweet of the first cohort. Again, focuses on safety, focuses on doing it the right way, and any questions we can address at the end of the webinar. But I appreciate it. Thank you for the opportunity. And the next speaker is going to be Kelly.
Thanks, Brian. Elizabeth, you can go ahead and go to the next slide. So as Karen introduced, my name is Kelly Dalby, and I have a son. He's in this picture. His name's Connor. He's currently 13 years old, and he has an SCN2A mutation. And when Connor was just a couple days old, he actually started presenting with very specific symptoms. When he was 8 months old, it's when he had his first seizure. And we went through a barrage of different tests. However, he went undiagnosed until he was 5 years old, and then he was diagnosed with an SCN2A mutation. And I really had like a laser focus that we had to find a diagnosis for him. If we understood the problem, there'd be a solution. And I was really very shocked when he was finally diagnosed through exome sequencing with an SCN2A mutation. We were told, we don't know much about SCN2A, and we have really nothing to offer you. And they apologized. And that was not at all how I saw his diagnosis going in my mind and what I hoped to achieve. And so really with the diagnosis, started calling scientists in the area, started connecting with as many people as I could. And I ended up starting a biotech company, and we named it RajCon. I was a co-founder among 2 other co-founders. We named it after my son, Connor, and another parent's son, Roger. And we were really just focused on trying to discover treatments for SCN2A mutations. And that's where PRAX-222 originated. And Praxis ended up licensing the program quite a few years ago, and all 3 of us co-founders now work at Praxis. And we started this program 8 years ago, and I cannot tell you how incredibly exciting it is to me and how emotional I have been lately at work. That we have IND approval. We have approval from the FDA to go into early onset gain of function patients with PRAX-222, which I had a hand in starting 8 years ago. And, we have reason to believe this molecule has potential to be disease modifying. So I'm just really excited for the SCN2A community and the potential that this has, and so grateful that, my son inspired me to have a hand in a huge team of people that is bringing this medicine to patients. So you can go to the next slide, Elizabeth. And so at RajCon and at Praxis, we really had an urgent need to have a data set on SCN2A patients to understand the burden of disease, to inform protocol design, to really inform our clinical development plan. And there was a retrospective natural history study happening in SCN2A, which we actually started at RajCon, Praxis sponsors. However, it was a 5-year study like traditional natural history studies, and we needed the data immediately. And so also some of the issues with just relying on a traditional 5-year natural history study, because what I'm about to go into, the citizen data, that can really be used to enhance these type of natural history studies. But without the citizen component, it's really a lot of burden on the caregiver already caring for a child with a DEE, or in this case, SCN2A. And then we're supposed to go out and collect all of these medical records. And depending on the age of the child, it's usually multiple institutions, it's dozens of EEGs. It can be up to thousands of pages of medical records, like for my son, for example, who's 13. And then clinic visits, if there's a prospective arm, more EEGs, surveys, assessments, and then you're relying on memory. And so if you have a son like I do who's 13, and I'm thinking back, talking to the physician, relying on memory about my son's early medication use and early procedures and early seizure history. One example that happened to me in my specific case is that one of my son's most prominent symptoms in SCN2A is a really specific movement disorder called choreoathetosis. And if we were relying on my memory, I would have thought he presented with that movement disorder well after the onset of seizures, at a couple years of age, up to maybe age 3. And whether a certain symptom happened prior to the onset of seizures or after is really understanding and knowing when you're studying the natural history of a specific disease. And so I answered in this natural history study that he presented with choreoathetosis at about 3 years of age, after the onset of seizures. And when I got all of my son's medical records and extracted data through Ciitizen, he actually presented with choreoathetosis at 4 months of age, well before the onset of seizures. And this is today still his most prominent and challenging symptom. And so really important to have data from those medical records that are comprehensive, extracted properly, to aid with relying on the caregiver's experience and marrying those two data sets together for really a complete picture. And so at this time, like I said, we really needed a data set very quick to inform our clinical development plan for PRAX-222, for PRAX-562, and we have another SCN2A program for loss of function patients also in early clinical development. And so I was introduced to Nasha, who works at Ciitizen, and I listened to a capabilities call where they described the Ciitizen platform. They used a different disease indication on their presentation, and it was really informative. I was really intrigued, but I really did not think that it was what we needed in the SCN2A space. I thought SCN2A was really much too complicated, and I did not think all of the necessary data could be found in the medical records. And Nasha was very confident that she felt it was. And so what we did is we made a deal, and I consented my son into the Ciitizen initiative, and they would take a few months, and then they would present all of his natural history study data to an entire team at Praxis. And so I consented him in. It really took about 5 minutes, not much longer than that at all. And one of the things I had to do was write the different institutions he was seen at. And a couple days later, I got my first email from Ciitizen that asked if there were 2 other institutions and named them that he was seen at. And in fact, he was during his first year of life. I had already forgotten that. And so they were able to find medical records that I hadn't even remembered at institutions. And then basically, in other natural history studies and in so many other research initiatives, we're asked as the caregiver to basically take a lot of developmental assessments or bring our child for developmental assessments. And one is the Vineland. I cannot tell you how many Vinelands I've taken on my son. It's always depressing. It's never fun. But in the Ciitizen extracted data, literally it's every single developmental assessment the patient has ever had with the raw score attached. So there's really no more need to keep taking developmental assessments. As long as you're refreshing your data every 6 months, it's up to date on the developmental assessments and the scores. And then the most important thing I can stress here, and why I love Ciitizen, the speed at which they're able to deliver natural history studies. For our SCN2A first cohort, From the time the recruitment closed to where we were able to receive the extracted data, 6 months, a lifetime of natural history study, data we were able to utilize immediately. And then this is an open access data platform. That means this is not Praxis's data. This is the patient's data set. Praxis is paying Ciitizen to go out and collect medical records on the patient's behalf with the patient's consent to extract all of the pertinent data from those medical records to then de-identify it and have it available for anyone that wants to utilize this data. And so Praxis has obviously licensed the data. Any other biotech company or pharmaceutical company in the space can also license this exact same data set, in addition to any researcher with an IRB approved study in SCN2A can also apply to Ciitizen to access the data. So it's so important. I mean, if I take my Praxis hat off, I want a medication for my son and for all these SCN2A kids. So I want everyone to have access to the same data set for equal opportunity to be able to do that. Elizabeth, can you go to the next slide? And then this is just, I'm not going to read word for word, but just 2 examples of an SCN2A parent, and we also have an SCN8A cohort in Ciitizen, but just giving examples of how the Ciitizen parent portal really has helped them. And so one experience was utilizing the data when they took a trip to the ER, and then a second one was being able to get insurance approval for a drug that they were unable to do so previously from being able to access the data in their parent portal that Ciitizen offers. And you can go ahead and go to the next slide. And so the Ciitizen cohort, the first SCN2A cohort, we ended up having 45 patients that enrolled. And with this specific data set, we used the 15 early onset patients of the 45 directly in our IND for PRAX-222. So this data was submitted directly to the FDA to help us paint the burden of disease. And then we also use this data to inform our protocol design, because we had to really quickly have answers to questions like, what's the age of seizure onset? How many seizures are these patients having? Do they remit at a certain age? What other comorbidities? What other medications are these patients taking? And this data set held the answers to all of those questions for us. And then also to advance and share with the broader scientific community. And so we actually had an AES abstract accepted for the data from this SCN2A cohort. So we'll be presenting at AES, we'll have a poster, and we're hoping to have a publication come out early next year for our 2A cohort, and then also our 8A cohort as well. And then this quote is from Steve Petru, our Chief Scientific Officer, and it was really, there was a press release out between Praxis and Invitae, as it was the first time the FDA accepted real-world data evidence in an IND, and that was with PRAX-222. We've also used the Ciitizen data in PRAX-562 documents as well. You can go to the next slide. And then I wanted to show a few data slides that we use directly in our IND submission for PRAX-222. So I'm just going to start with the early onset patients. So like I mentioned, this is 15 early onset patients. And on the left-hand side is the age of the patient at the time that we received the records. And in the literature, it talks about early onset patients tend to have frequent seizures early in life, and they tend to remit. And what we saw in this Ciitizen cohort of early onset patients is all of these patients, except for one, continued to have seizure activity until the end of records, which is denoted by this black line on the right-hand side. And so that meant for us that we were able to use seizures as our primary endpoint in the PRAX-222 trial. And then another thing to point out is status epilepticus. So in green, those are the green dots. And this is really a life-threatening emergency situation where the seizure just does not stop. And 10 out of 15 of these early onset patients experience status. And as you can see, some of these patients, multiple episodes, and in one patient throughout a lifetime. So it really paints the severity of this disease and shows the burden of disease. And then you can go to the next slide, Elizabeth. And then this is really showing the impact of the disease. Just for early onset, we're looking only at the first year of life. And patients tend to present with seizures when it's early onset, the first 3 months of life, but the majority of these patients in the first few days of life. And so 14.5 medications prescribed on average in the first year of life alone. And so what is this telling us? This is telling us these medications are not good enough. And it's not just the medications used to try to control seizures or to try to reduce seizures. It's several different medications used for other comorbidities, like behavior, like anxiety, like sleep, like feeding issues. And so these patients are really trialing multiple medications in the hope for symptomatic relief. And as you can tell with this high count, we can assume they're just not helping enough. And then procedures, so therapeutic and diagnostic procedures. Again, I have to stress in the first year of life, so first 365 days, 21 different procedures that the patients went through. And in this patient population, it's things like EEGs and CT scans and MRIs and feeding tubes surgically implanted. And then we have hospitalization, so on average 4.3. But if you look at the duration, 70 days spent in hospital. So that's roughly 20% of the patient's time and essentially the family's time spent in the hospital in the first year of life. So very severe, very burdensome disease. And again, these statistics were absolutely necessary to use in our IND filing to paint this picture, the accuracy of the disease and what these patients and families are experiencing. Elizabeth, next slide. And then this includes other patients in addition to our early onset, but this is really time from first seizure onset, which is the green line, to SCN2A genetic diagnosis, which is the circle. And as you can see, these older patients, obviously many years went from time from seizure onset to genetic diagnosis, contrasted by these younger patients, more present day, where they're diagnosed almost immediately or within days to months of having their first symptom of SCN2A disease to SCN2A diagnosis. And my son falls into this earlier category, like I mentioned. He's currently 13. And when he presented with his first seizure in 2010, we had a genetic epilepsy panel. It came back normal because SCN2A was not added to the panel at that time. And that didn't happen till about 2014, 2015, 2016, when it became more standard to administer genetic epilepsy panels. And at that time when it was administered, there were a couple dozen genes on that panel. Compared to present day in 2022, there's over 300 genes known to cause pediatric epilepsy on that panel. And so a lot of these older patients, someone had to be advocating for them to go back and repeat the panel or to get full exome sequencing. And so there are probably, we can assume, a lot of patients out there that are older that did not repeat a recent panel, that did not get whole exome sequencing, that are still out there without an SCN2A diagnosis. And this becomes really important when we're talking about companies like Praxis making medications that are targeted to the genetics, so like PRAX-222. And then you can go to the next slide. And so in order to present some more of the data, which is patient populations beyond, or I guess phenotypic buckets that extend past early onset. So every single variant of every patient in our SCN2A cohort, we had that variant characterized using dynamic clamp to determine if it was loss of function or gain of function, mixed function, or it had no change to wild type. And so our early onset patients, all of these patients ended up having a gain of function mutation. And typically, there's always going to be outliers, but typically seizures present in the first 3 months of life and usually in the first few days of life. And sodium channel blockers tend to be or can be beneficial in this patient population. And then our next group, we're calling late onset infantile spasms. And these are patients where their first seizure presentation is always infantile spasms, and then they go on to have other seizure types. These infantile spasm seizures present on average between, let's say, 6 to 12 months of age. They have been seen up to 18 months of age. And for some of these patients, sodium channel blockers can worsen their seizure frequency and severity. And the ones that we've done the characterization work so far are loss of function. Again, I'm going to repeat, there are going to be outliers. Not every single patient with an SCN2A mutation is going to fall perfectly into this bucket. And then our next categories we're just calling late onset. Typically, seizures present between 18 months to 4 years of age. We've seen them all the way up to 12 years of age for first seizure presentation. And first seizure presentation is a seizure other than infantile spasms. And same thing where sodium channel blockers can worsen seizure frequency and severity. These tend to be loss of function. And oftentimes, autistic-like characteristics can present prior to seizure onset. And then our fourth group is these patients have autism. Many of these mutations are nonsense, so known loss of function. Some are missense. And a lot of these patients can have developmental delay or developmental impairment or autism, and to date have not had a seizure. You can go to the next slide. So our original cohort was 45 patients, like I mentioned. 22 of those were female, 23 were male, so we got a nice even spread. And then our ages ranged. Our youngest patient was 8 months old, and our oldest patient was 23 years old, and the average age was 7. And then we also got a really nice even spread of the different phenotypic buckets. So we had 15 early onset patients. We had 9 patients that had late onset infantile spasms. We had 10 with what we're calling late onset epilepsy. And then we had 11 with autism only, or patients with autism that have never had a seizure. Next slide. And then the mean days to seizure onset. So in our early onset group, average age to seizure onset is 5 days. And then late onset infantile spasms, 380 days or just over a year. And then late onset, 807 days, so just over 2 years, 2 years and a few months. Next slide. And then this slide is basically seizure history from time of birth to present. And I showed you something similar in the early onset population. But if you look at this dotted line again, this signifies 3 months of age. And the beginning is all of our early onset patients. And so again, all of these patients presented with seizures other than infantile spasms prior to 3 months of age. And then our next group moves into late onset infantile spasms. And all of these patients presented first with a very specific seizure type, infantile spasms, which usually involves hypsarrhythmia on EEG. And then after that is our late onset population. So seizures other than infantile spasms at onset. And as you can see, all of these phenotypic groups, many of these patients have had bouts with status epilepticus. And in our early onset group, there are several patients that also had a bout with infantile spasms. It was just not their seizure onset that they presented with. Next slide. And then this slide is really interesting to me. So this is comorbidities seen in SCN2A. And we know as parents, seizures are one symptom among many really devastating symptoms. And so what was really interesting to me is regardless of gain of function or loss of function, the majority of these patients have very similar comorbidities, whether you're late onset infantile spasms, early onset, or just autism only. And some of the most common other comorbidities in addition to seizures, so developmental delay or developmental impairment, hypotonia, decreased muscle tone, a lot of patients having sleep disorders, and so really waking up frequently throughout the night, having issues staying asleep, some patients going days without sleeping, a lot of GI problems, so GERD, feeding difficulties, needing feeding tubes surgically implanted. So again, seizures, one symptom among many devastating symptoms, and then also helping us inform what other exploratory endpoints are we going to add to PRAX-222? What else do we need to see improve in addition to seizures? What else is important or burdensome to families? And I mentioned for my son, yes, of course, seizures, but choreoathetosis, a movement disorder. very, very devastating, impacts him incredibly with regard to development. And so for me, that would be really important that I'd want to see improvement in addition to sleep, in addition to GI, and in addition to seizures. Next slide. And then total medications per individual. And so in gray is our medications given for comorbidities other than seizures. And then in blue is AEDs that are not in the sodium channel blocking class. And then in the lime green is sodium channel blockers. And on the left-hand side, it gives you the phenotypic classification of that patient. So either early onset, late onset, autism only. And what we can see is there is a heavy burden on medications trialed in this patient population. And then this is also the age right before the colored bar graphs of the patient at time of extracted data. So you can see even very young patients all the way up to older patients trialing through dozens of medication, with one patient trying 56 different medications in total lifetime. And so again, just an assumption, these medications are not good enough. They're really targeted at symptom relief, and the majority of the time not doing a good enough job. And so the slide I showed you earlier on early onset medication usage, now we can see it's also the same in the different phenotypic buckets as well. So really urgent need in this space to develop disease modifying therapies for this patient population. Next slide. And like I mentioned, our first cohort was 45 patients, and this data was really incredibly valuable, not only to Praxis, but like I said, it's open access data, so other researchers can utilize this data, other biotechs making medicines for SCN2A patients. And so this QR code right here, you can use this right now to sign up for our new 100 patient cohort of SCN2A patients. We're really looking to enroll any SCN2A patient. So early onset seizures, late onset seizures, if you have autism without epilepsy, intellectual disability, really any patient with an SCN2A diagnosis. And just wanting to highlight again, really all of the benefits of Ciitizen. So institutions only have to save medical records for about 7 to 10 years. And then they don't have to save that data anymore. And that's something that I learned in the process of setting my son up with Ciitizen. And so I lost some of his early EEGs and some of his early medical records. And that information is invaluable to certain researchers or biotech companies in the drug development process, especially those early EEGs. And so one reason is to go out and have all of your medical records and EEGs and other tests collected. It's in a parent portal. You can utilize that and it will never disappear, even when the institutions don't have to keep them anymore. And then, like I mentioned before, just wanting to stress the open access data platform. And it's no burden to the parent or the caregiver, it's only added benefit. So I utilized my Ciitizen portal and sent all of his medical records for the retrospective natural history study in SCN2A. I've also used this portal to share his medical records with another physician for a second opinion for a symptom he was having. And I've had other parents, I shared a couple quotes, but I'm often told stories of how caregivers are utilizing this portal. So tremendous benefit to the caregiver and patient, in addition to aiding in research, in addition to the open access data platform, where it's not Praxis's data. I just need to stress that. And really, I can't think of one reason not to join Ciitizen. I've tried and I can't think of one reason not to. I really think it's imperative to. So anyone that's already been a citizen or a patient in our first 45 patient Ciitizen cohort, you can go ahead and refresh your data as we open this cohort a little bit less than 2 years ago, so that we can get the most updated records from the last couple years. And then anyone that hasn't signed up, we just urge you to do so. We're making it very simple for you to do with the QR code here, or you can also get information, of course, on scn2a.com. And then I think that's it for my part.
Thanks, Kelly. Like you really conveyed, it's so exciting to see where the participation and the contributions of the community have gone with the study. And thank you to everybody who's participated. That includes thanking everybody who helped motivate each other in the community to help contribute to growing the disease understanding and the understanding of the daily life and the life challenges of families living with SCN2A-DEEs. So just to recap, the EMBRAVE study of PRAX-222 is a potentially disease-modifying antisense oligonucleotide for SCN2A-DEEs. We'll be starting with an initial pre-qualified small cohort in the United States in the coming months. Simultaneously, we're exploring ways to initiate other cohorts in different geographies. I think also to recap what we've just learned from the Ciitizen program as well, there will be about 100 spots in the next Ciitizen cohort. So we encourage people from representing kids with all different SCN2A-DEE backgrounds to be able to participate in that. And of course, the more people who reach out to us for the EMBRAVE study of PRAX-222 through the EMBRAVE study email address, which we'll put up on the screen now, we want to make sure that we have all of you signed up and ready to hear more about the coming weeks and months of these cohorts going live. So if you're interested to participate in the EMBRAVE study immediately, you can email us and we will start the conversation rolling there. And of course, sign up to Praxis Communications. All you have to do is hold your phone up and use the camera to scan this QR code. If you need the Ciitizen link as well, we can also send that to you as well. But by all means, you can always email us directly. And finally, next slide, please. If you have any other questions, you can always reach out to us at patientadvocacy@praxismedicines.com, where Karen, myself, Kelly, whoever from the team you have your question most pointedly for, we'll make sure it gets answered as best we can. Please understand, as Karen said at the beginning, there will be questions we don't have answers for right now, but it's because of where we are right now. And more questions and answers will come as more announcements unfold over the coming weeks and months.
We want to thank you all again for joining us. Thank you for your time this evening. And we will proceed with updates as we can. And I guess
look forward to seeing you.
We look forward to seeing you. Thank you.